Okay, so now for today, this is our third part of our first online lecture. We just finished in our part 1 and part 2 some basic introduction about the viruses, and in this continuing section, I will be discussing a little bit about the prokaryotic cells, especially with reference to the protozoa. We all know that we divide the organisms, or you can say all the living creatures, in two wide groups: prokaryotes and eukaryotes. Prokaryotes are the single cellular organisms, while on the other hand, eukaryotes are all those who are multicellular. Among prokaryotes, we have our bacteria, viruses, protozoa, um, prions also we consider under those that are as unicellular. On the other hand, eukaryotes, we humans, animals, plants, they all are part of the eukarya. If you look for fungus, so fungi are divided into two broad groups: yeast and molds. Molds are considered under the eukaryotic cells. They are multicellular, while the yeast cells, these are the single cellular organisms, and we often consider them under the heading of the prokaryotes. The size of prokaryotic cells led to their being undiscovered for most of the human history. In 1546, there was an Italian physician, Girolamo Fracastoro. He suggested that diseases were basically caused by unseen organisms, and for these unseen organisms, right now we are using two important technologies. That is, one is the microscopy, by observing them using different type of microscopes, light microscope, electron microscope, and so many others. And on the other hand, we can also identify and look for the presence of these infectious agents or the microorganisms on the basis of presence of their molecular particles, especially their DNA or their proteins. If we look for the history of microbiology, so the first name that comes to our mind is of Leeuwenhoek, Antoine van Leeuwenhoek. He was the first person. Basically, he was a Dutch linen merchant who was used to look for the quality of different type of clothes by using different type of single lens hand lenses, or the single lens microscopes that we commonly call as hand lens. And he was in hobby of making these hand lens, and he made the hand lenses that can see the things up till 500 magnification. So beside his textile merchant activities, he also used to observe variety of different specimens just for hobby. For example, he used to observe that what's there in the water drops, in the in the different type of other secretions. For example, saliva. For example, tears. For example, sweat, feces, and all these type of the raindrops. And what he find in all these different observations, that there are some particular tiny creatures that are present in all these samples, and he gave them a name. Animalcules means tiny animals. So he is considered as the father of microbiology because of his observations and his first reported drawing about these creatures. Then another name that we are mostly familiar with is Louis Pasteur. He refuses the idea of spontaneous generation. What spontaneous generation is, that before him there were two main groups. One group was supporting the idea of spontaneous generation, that the living things can arise spontaneously from other living things, while the other group, that was a opposing this idea, they said that no, there is no need of any living thing to create the new living thing, or the for the living thing to arise from. So basically, Louis Pasteur, he used his famous swan neck experiment, in which he used the flasks, glass flask of various shapes. Basically, the bottom of the flask was same, but the neck area of those flasks was different, and and he used these flasks to show that if the broth that is present inside that flask is exposed to the open air, it will get rotten. But if it is not exposed to the direct air and it goes through a process, the air goes through a process while it reach the broth inside the flask, it might save the broth from getting rotten. And by having this swan next experiment, he proved that for the existence of life, the presence of life is very important. Then the name of Robert Koch is one another important name that we are all familiar with reference to microbiology. He gave us the theory of germ theory of um, microorganisms, that all the diseases, all the infections, they are caused by a specific type of microorganism. Like he studied, basically he started with the study of anthrax bacilli that causes anthracis in the sheeps, and he proposed four postulates to prove a causal relationship between a microorganism and a disease. And what he suggested, that for example, if a sheep is having anthrax, then the causative agent should be the anthrax bacilli, that is Bacillus anthracis, not the E. coli or some other microorganism. So the important thing is that what he proved through a series of experiments, that behind every single disease there is a specific microorganism, and this is these four postulates of Robert Koch, they are familiar, or they are famous with the name of the germ theory of diseases. Now, if you see here this picture, this is a pipe, okay, and you can see here it is covered with these blue colored microorganisms. So it is just an example of a biofilm. Now you can ask me that what biofilm is. So just think of your bathroom curtain. So what happens that gradually when you change a new curtain, it is clean, clear, fresh, and when the days passes, you will observe that gradually it will become slightly moist. A layer, a very fine layer of moisture is there always, or no matter how you clean it, but still you can find this fine layer. Basically, this is when the moisture is there on that curtain. Gradually, the environmental bacteria starts to accumulate or get attached to that curtain, and gradually, like they gather more organisms, and a very fine layer of microorganisms develop that are attached with each other by means of their capsular material, their slime material, and this layer of the microorganism, it is called as biofilm. So when we look for the prokaryotic diversity, the oldest, structurally simplest and most abundant form of life that we find is the bacteria. They were abundant for over a billion years before eukaryotes or the multicellular organisms were appeared. 90 to 99% of these are still unknown and undescribed. They fall into two domains, that is bacteria and archaea. And many archaea, these are the extremophiles, that whether they will live into the extreme high temperature, extreme low temperature, extreme high concentration of salts or sugars or so. Now, if you look for the specific characteristics of the prokaryotes or the unicellular organism, the most important thing is that they are unicellular. Most are single celled, may stick together to form association, just like we see in the previous slide in the biofilm. Their size varies tremendously, and most are less than 1 micrometer in diameter. With reference to their nucleic acid, chromosome is single circular double-stranded DNA, and it is found in the nucleoid region of the cell, and most of them, they do not have a true nucleus, and some of them might also have plasmid. These are the some extra genetic material that we can find in some of these prokaryotic cells. How they divide? They divide by means of binary fission, means one cell divides into two, two into four, four into eight, and so on. When we talk about the genetic recombination, the exchange of the genetic material extensively through horizontal gene transfer happens, and basically it is not a form of reproduction. If you look for the internal structure of these prokaryotes, so you will see that there is no membrane bounded organelles are found, like the nucleus is totally absent. The nucleic acid is found dispersed throughout the cytoplasm. No internal compartments are present, and the plasma membrane can be extensively enfolded. Flagella can be found in some of these prokaryotes. They are simple in structure, they are different from the eukaryotic cells flagella, and again the function of these flagella are to help them in movement or motility, but not all prokaryotes have flagella. Metabolic diversity, if you look for that, how they utilize different type of metabolites present in their environment. They can be oxygenic or anoxygenic, means they can perform these metabolic activities in presence of oxygen or in absence of oxygen. Some of them can also perform photosynthesis in the presence of sunlight, and they can utilize these metabolites in presence of the sunlight. Some Some also use variety of chemicals present in their surrounding for their routine metabolic activities. Now, when we say there are two basic domains that we have the evidence that they were found in the universe since so long, these are the bacteria and archaea. Basically, how they are different. There are four key areas that makes them different from each other. Number one is the plasma membrane, then their cell wall compositions, the way how their DNA replicates, and then how their genetic expression take place. So basically, the scientist Carl Woese was the one who determined this three-domain system of phylogeny. And in this three system of phylogeny, he says that they all three arises from a common ancestor, and these three domains are the bacteria, archaea, and eukarya. So when we say plasma membrane, all prokaryotes have a plasma membrane. The other name that we use for the plasma membrane is cytoplasmic membrane. These membranes, when they are in archaea, they are different from the bacteria and from the eukaryotes. Basically, the archaean membranes are formed of glycerol that is linked to the hydrocarbon chains by ether linkages, not the ester. While in case of bacteria and eukaryotes, these linkages are chemically ester in nature. The hydrocarbons that could be found in the archaea, they could be branched, and the tetraethers forms a monolayer instead of a bilayer. like we know in case of the bacterial cytoplasmic membrane, we all are aware of those phospholipid layers. So it allows the extremophiles to withstand the high temperatures. If we look for the cell wall, all prokaryotes have the cell wall. Bacteria have important ingredient, that is peptidoglycan, and the gram-positive bacteria, they have a major content of these peptidoglycan. Gram-negative bacteria also have peptidoglycan, but the content is very less. It is about approximately 1%. On the other hand, the Archaea, they lack peptidoglycan. They does not have peptidoglycan at all in their cell wall. If we look for the DNA replication, both Archaea and the bacteria, they have single replication origin, nature of origin, and protein used are different. So basically, the archaeal DNA replication is more similar to that of the eukaryotic cells. If we look for the gene expression, archaeal transcription and translation are more similar to those of the eukaryotes. Now, if you remember, transcription and translation, they both are the first two steps in case of protein synthesis, that how the DNA translated into the messenger RNA, and then messenger RNA transferred the information that it gets from the DNA towards the ribosomal RNA, and then with the help of the ribosomal RNA, transfer RNA, and the ribosomes, the organism or the cell starts to perform the protein synthesis. With reference to the early classification, there are a variety of characteristics that were considered. Prokaryotes not easily classified according to the forms. We have variety of the forms, and early system, it was basically relied on staining characteristics and observable phenotypes, means like the phenotypic characteristics. So either they can be photosynthetic or non-photosynthetic. They can be motile or non-motile, unicellular, colony forming, or filamentous. We can also look for the formation of endospores or exospores, or that how they divide either by the binary fission or something else. And most important is that are they causing any kind of infection in humans or not. So these all are the different phenotypic characters that were considered first or in the early days of the process of classification of these prokaryotes. On the basis of their molecular composition, we can also classify them amino acid sequences of key proteins, then the percentage of guanine and cytosine, or you can say GC ratio, always play important role in the molecular classification of the microorganisms. Nucleic acid hybridization is also another way of classifying them. It is closely related species will have more base pairing as compared to the one that are not related. Gene and RNA sequencing, especially ribosomal RNA sequencing, is currently a very popular method of helping in molecular characterization or the molecular classification, especially the 16S ribosomal RNA. And beside that, like we have the procedures available for the whole genome sequencing. Based on these molecular data, several prokaryotic groupings have been proposed. Just like we all use in our English literature different type of dictionaries, similarly in biological like this microbiology, and for the to understand the classification of the bacteria, Burgess Manual of Systemic Bacteriology is considered as the Bible of the microbiology that provides all the details about all these microorganisms. Large-scale sequencing of random samples, it indicates vast majority of bacteria have never been cultured or studied in details. So most of the time the importance is given to those bacteria that are common and are of main concern with reference to their pathogenic or any other special characteristics. So if you see here, you can see that we have different groups of these prokaryotes with reference to their sizes. So here is the Archaea. They differ greatly from the bacteria. Here you can see all these bacteria here. So you can see they are totally different. Although both are prokaryotes, Archaea's cell wall lack the peptidoglycan. Plasma membranes are made of different kind of lipids than the bacterial plasma membrane. RNA and ribosomal proteins are more likely eukaryotes than bacteria, and mostly they are anaerobic. Excuse me. For example, like the examples of these archaea are Methanococcus bacteria, Methanococcus, Thermoproteus, and Halobacterium. And then, if you compare this with the other bacteria, for example, Aquificii, it represents the deepest or oldest branch of bacteria. Equifax pyrophilus is a rod-shaped hyperthermophile. Hyperthermophile means they can survive at very high temperatures with a temperature optimum at 85 degrees centigrade. It is a chemoautotroph, means it can utilize the chemicals present in the surroundings, and it can also use the carbon dioxide as a sole source of carbon. It oxidizes hydrogen or sulfur, and several other related phyla are also thermophiles. So it is a thermophile that love to grow and survive at the high temperatures. And then we have the bacilli, gram-positive bacteria, largely solitary. Many of them they form or produce endospores. They are responsible for many significant human diseases. For example, Bacillus anthracis that causes anthrax, Clostridium botulinum that causes botulism, and then we can also see strepto uh, like Clostridium tetani that causes tetanus, etc. And you can see the size is around like 4 micrometer. We have some gram-positive bacteria, actinobacteria are the example. They form branching filaments, and some produces spores, and sometimes we take them or mistaken them as fungi. Although they are bacteria, they produce many commonly used antibiotics. For example, streptomycin, tetracycline, one of the most common type of soil bacteria, also common in dental plague, is Streptomyces actinomyces. Spirochetes are another example of bacteria. They are long, coil-shaped cells that stain gram-negative. They are very common in aquatic environments, and rotation of internal filament produces this corkscrew-like movement. Some of these spirochetes, such as Treponema pallidum, causes syphilis, and Borrelia burgdorferi, that causes Lyme disease, are significant human pathogens from these spirochete groups. Now, if you look for a little bit more detail here, that these Crenarchaeota and Euryarchaeota, they are from the Archaea, while Aquificii, Thermotogiae, Chloroflaxiae, Deinococcus thermus, they all are the thermophiles that love to grow at high temperatures, while the Bacilli, Clostridium, Actinobacteria, they all are the gram-positive bacteria. Um, these spirochetes, as we have mentioned here, they are gram-negative, or they are different, slightly different in shape, like they look like spirals. Now, if you see here in this picture, uh, we have cyanobacteria. These are another form of photosynthetic bacterium, means they can perform photosynthetic activities, and they can make their own food by using the sunlight. They are deeply pigmented and often responsible for blooms In polluted water, both colonial and solitary forms are common, and some filamentous forms have cells that are specialized for nitrogen fixation. And the size you can see here, they are around 10 micrometer in size. Beta cells, these are nutritionally diverse group that include soil bacteria like Nitrosomonas that recycle nitrogen, and they are very good for our fertilization process in the crop fertilization. And the gammas are a diverse group including photosynthetic sulfur bacteria, and they might be pathogens, for example, Legionella, and enteric bacteria that inhabit animals' intestine. Enteric includes Escherichia coli, or in common terms we call them E. coli, Salmonella that causes food poisoning, and Vibrio coli that causes cholera. Pseudomonas are a common form of soil bacteria, and they are responsible for many plant diseases and are important opportunistic pathogens. In delta group, we have the cells of myxobacteria that exhibits gliding motility by secreting slimy polysaccharides over which masses of cells glide, and when the soil dries out, these cells aggregate to form upright multicellular colonies called fruiting bodies. If you look for the examples of these bacteria, Vibrio and Geobacter are very common bacteria that also help us in the bioremediation processes. If you look for the shapes of these bacteria or the prokaryotic cells, there are three very common basic shapes. Bacillus. Bacillus means these all are the Greek words. So bacillus means rod. So these are the rod-shaped bacteria. Fucus means round or spherical. So these are the spherical shaped bacteria. And then the spirillum or the helical shaped bacteria. So these are the three very common shapes that you can see in the prokaryotic cells. Their cell wall, as I told you before, that they have peptidoglycan. Now this peptidoglycan can be less or more in different groups of bacteria. They form a rigid network made up of two important sugars, N-acetylmuramic acid and N-acetylglutamic acid, and these sugars they are interconnected with each other by means of a special linkage that is called as beta-1,4 linkage, and this is one of the important structure found in the cell walls of gram-positive bacteria that helps us in identification of these bacteria or differ them from the gram-negative bacteria that does not have that much amount of the peptidoglycan. It maintains the shape, it withstands the hypotonic environment, and Archaea have a similar molecule that is called as pseudomurine but not the peptidoglycan. So this is the difference between the bacteria and Archaea, that bacterial cell wall has peptidoglycan while the Archaea cell wall has pseudomurine. In 1845, there was a scientist named Hans Christian Grahn. He looked for the bacterial cell wall structure, and on the basis of the bacterial cell wall structure, he derived one method of staining that help us to differentiate differentiate the bacteria into two big groups. One is known as gram-positive bacteria, and the other group is known as gram-negative bacteria. So the gram-positive bacteria, they have a thicker peptidoglycan wall, and they stain a purple color, or they take up the color of the primary dye, that is crystal violet, and it is violet or purple in color. While on the other hand, the gram-negative bacteria, they contain less peptidoglycan and do not retain the purple colored dye. And because during the procedure, when we treat these cell walls with the iodine, and then later on with the alcohol, that is 75% alcohol, what happened that all the lipid content of the cell wall of these bacteria get dissolved in the alcohol, and as a result they lose the color. So they can take the color of the counter stain, that is saponin, and it looks pink or reddish. So here you can see in this slide the process of gram staining that we commonly use to differentiate the bacteria into two big groups, that is gram stain used to differentiate into gram positives and gram negatives. So what happens that first we make a very thin film of the bacterial cell on the slide, and then we apply the first or the primary stain, that is crystal violet. After that we apply a mordant, chemical mordant, that is iodine, and then we wash this uh, film with the alcohol, 75% alcohol. So those bacteria that have more of the peptidoglycan in their cell wall, they will keep this violet color as it is, while those that does not have that much amount of peptidoglycan in their cell wall, and instead they have more of the lipid in their cell wall, the lipid will get dissolved in this alcohol. Now what will happen, that because of this solubility of the lipid, the color will be lost, so they will become colorless. And then when we apply the secondary dye or the counter dye, that is safranin, and it is red in color, the colorless gram negative bacteria will take up the color of this secondary dye and they will appear red or pink in color. So that is why at the end of this gram staining we will have two colors bacteria. One bacterial group will be appear as purple or violet, while the other group of the bacteria will appear as pink or red. And that is how we divide the bacteria into two big groups: gram positive bacteria, that is purple or violet, and gram negative bacteria, that is pink or red, on the basis of their cell wall composition. Again, just to remind you that those that are rich in peptidoglycan, they will appear violet or gram positive, while those that have less peptidoglycan and more lipid content, they will appear pink or red, that is gram negatives. Now if you can see this here a little bit more details, so you can see the difference of the cell walls of both group of bacteria. So here this is the gram positive bacteria cell walls. You can see the thick layer of the peptidoglycan, and then this is the cytoplasmic membrane, which is a phospholipid bilayer. On the other hand, if you see here, the peptidoglycan is just this much, while rest of the layer is of the lipids. So that is what happened when we apply crystal violet. So this crystal violet will remain trapped in the peptidoglycan here in case of gram positives, while here the outermost layer is lipid. So when this thin film is going to be treated with alcohol, alcohol will dissolve this lipid layer, and as a result they will lose the color, and this is how they will appear colorless. And when we apply the second counter stain, it will color this peptidoglycan layer that is innermost, and that's how they will appear pink or red in color. So the gram-positive bacteria, they are thick complex network of peptidoglycan. They also contain lipoteichoic acid and teichoic acid. While on the other hand, the gram-negative bacteria, they are thin layer of peptidoglycan, second outer membrane with lipopolysaccharides, and they are resistant to many of the antibiotics. Why? Because most of the antibiotics, they target basically the peptidoglycan. So if the peptidoglycan content is less here in the gram negative cell wall, and also it is not exposed outside, so the antibiotics will not affect the gram negative bacteria easily. Now what is S layer? S is for slime. So a rigid paracrystalline layer found in some bacteria and Archaea. Outside of peptidoglycan or outer membrane layer in gram-negative and gram-positive bacteria, this is an additional layer that has diverse function. It is, or most of the time it is involved in adhesion or attachment to different surfaces. Some bacteria have another quality of making capsules. What capsules are basically? Capsules are gelatinous layer of carbohydrates that is accumulated around the cell wall of these bacteria, and again it helps in the attachment to different surfaces, and it also makes the bacteria more pathogenic, because this capsule is gelatinous and it can easily escape the pathogenic phagocytosis mechanism of the host body, and that's how it protects the bacteria from our immune system. Bacterial cells or the bacteria, some of the bacteria might also have flagella and pili. What flagella are? Flagella are cylinder, rigid, helical structures, thin hair-like structures coming out of the bacterial cell wall, and they are composed of a special protein called flagellin, and mainly they are involved in the locomotion or the motility of the bacteria, and they spin just like the propeller. On the other hand, pili are also short hair-like structures coming out of the cell wall of the bacterial cell, and they does not take part in mostly the motility, but what their function is, they have some attachment and adherence to different type of surfaces. So that's how they increases the pathogenicity of the bacteria, plus they also help in the conjugation process. Excuse me. Now, if you want to see detailed structure of the flagella here, so this is a bacterial flagella. You can see here, this is the cell wall, this is the cytoplasmic membrane. So it is arising from the base of the cytoplasmic membrane, and then this is going out. Just if you think of your hairs or that are coming out of the skin. So it is comprises of these two rings, outer ring and the inner ring, and they are made up of a special protein called flagellin, and then finally the filament that is coming out, and this filament is connected with these rings with the help of this hook, and it just simply work like a propeller. What endospores are? Endospores are basically highly resistant dormant bodies that are produced by some of the bacteria. So remember, the capsule, flagella, pili, or endospore, they all are those structures that are not found in all of the bacteria. They can be found in some bacteria that carries the genes for their production. So endospores, they are the special structures that are developed by some bacteria that develop a thick wall around their genome and a small portion of the cytoplasm when exposed to the environmental stress. So you can say that in adverse conditions, when the survival is difficult for the bacteria, what they do, they make these highly resistant internal bodies called endospores, and when conditions improve, they can germinate and return to the normal cell division. And for example, the bacteria that causes tetanus, Clostridium tetani, is a spore forming bacteria. The bacteria that causes botulism, Clostridium botulinum, is a spore forming bacteria. And then anthrax is also a disease that is caused by a spore forming bacteria named Bacillus anthracis. So I'm just stopping here, and we will continue with the next next part. Thank you very much.